So today we'll talk a little bit about molecular signatures, which are measurements of molecules that you can use as a signature, you can hear from the name. So this is just one very clear example where you have, so this is from gene expression, so you measure the expression of on one group of genes is low in AML in this case, and usually high in ALL, but not always. And then another group of genes that's the opposite. But as you can see, there's a lot of noise. So if we would choose any one of these genes and try to make a signature, it wouldn't work very well. But together, they work really well. And this is, of course, what people have then seen the potential for this, that if we can measure lots of things, we can measure mutations, copy number changes, changes in gene expression, protein levels, phosphopeptides. If we can then put these together and select out signatures. The problem that we'll talk about later is that it's easy to measure a lot of things. And as we discussed last time, if we then have look at it unbiased and just try to find signatures out of this enormous number of measurements, we run into the problem that we do many tests. So we need to do correction for multiple testing. So it's not always an advantage to have just an unbiased sampling of as many things as possible. If we can use our knowledge to focus in on a few genes, that's much more, can be much more fruitful. But then, of course, we have to select the right ones to focus on. So there's a lot of interest, and so these are, you can call them molecular signatures or in vitro diagnostic multivariate assays, but there is definitely, it's very promising. So, and we can use them, apply them to many things. We can apply them to diagnostics. One part of diagnostics would be early detection, which is of course the dream, that to take a blood sample and long before a disease becomes serious, we would detect it. But this is usually very difficult, extremely difficult. And there are very few examples of this where it's successful. So, for example, the problem is that if you want to apply this to the general population, it will be you will have to have a test that really is both sensitive and specific, because otherwise you get too many false positives. So the best thing is to, if you can have maybe a hierarchy of different tests. So you have one that's cheap and easy to apply, and then that might have a lot of false positives. But then you have another test that's not so much more expensive to have another, and then have these hierarchies of tests. So, but it's researchers don't even want to go to early detection because so for example, one problem is also that if you have a tumor and you want to do early detection, the tumor is very small. And then let's say, and you want to look in the blood, so you're looking for proteins that leak out of this tumor as diagnostics, for example. So then you have a small tumor, and then it gets diluted in the whole in the blood. So then you get, the concentration is going to be very low. So it's that's one thing why especially the proteomics markers are very for early detection. But something that's probably much more promising is to use markers of personalized treatment. So when we know that someone is sick, we can do a test and then, based on that test, decide how the treatment should be done. And here there is a lot. So, for example, in cancer, often there are quite a few drugs available and a few different ways to treat at least the common cancers. But usually the doctors, they don't know which one to pick. So there could be a big potential of developing diagnostics to guide the treatment. So these, and there are other types of. But before we talk more about molecular markers, I'm going to talk a little bit about experimental design, because as we'll see later on, a lot of times the discovery, the experiments to discover molecular markers are not so well designed. So, and it's surprisingly easy to go wrong with experimental design. As you see, this is a cartoon that shows one example, but it's quite difficult to really think through all the steps in experimental design. So the main problem is that we have either by chance we can get a result that we believe in, and this is what we talked about with the multiple testing, for example, and that's where we want to sort of control that we by chance would get a result. But then the other big problem is bias, and that's where in both of these is where good experimental design comes in. So when we talk about experimental design, we can look at it as a system that has an inputs, and then it's a process and outputs, and then there are controllable factors and uncontrollable factors, and we want to try to then design the best experiments to sort of still get good results in the face of both the controllable and uncontrollable factors. So the main thing with experimental design is to sit down and take time to think about the experiment. And this is really starts with first thinking through what is that one wants to do. What's the question? Is it testing specific hypothesis, or is it a discovery exercise? Then one would select response variables, and then choosing controllable factors and what would be their normal range that one wants to investigate. And then also sit down and think about all the uncontrollable factors and estimating their effect. So things for mass spectrometer-based proteomics, uncontrollable factors would be, for example, the humidity in the room where the mass spectrometer is, the temperature of the room, and these can in principle be controllable, but sometimes it's really too expensive to control them, and so one can, in this case, by spending a lot of money, of course, make these uncontrollable factors controllable, And then if one it's worth it and one has the money, of course, then it's worth doing that. Then we choose an experimental design, perform the experiment, and then comes the data analysis. And then, of course, this is usually an iterative process that then we go on and design the next experiment based on our results from the first one. So then exploring parameter space. So if we have many different factors, we can try them to vary them within their range one by one. And then we would, in this, if we say that we want to try test the two extremes within the range, then we have 2 k experiments if we have k factors. So that's so that just grows linearly with the number of factors. But the problem is that we could have interference within the factors, so that if we would vary 2 at the same time, we would get very different results. So one could then think of a 2-factorial design where one tries for 2 factors, tries all the combinations. So that would be 4. If we then have 3 factors, we would have 8 experiments, and then 2 to the k experiments for a k factorial design, which very fast becomes a large number. So if you have 7 factors, 128 experiments, but 10 factors is 1,024 experiments. And so even a few factors is usually impossible to try all the combinations. So the other thing we should think about is randomization. And that's we want to randomize with respect to everything that we can think of. For example, which order we analyze things in, where we store different materials. Let's say we have different, if we do lots of experiments on cells, when we incubate them, we want to also randomize where they are placed, and so on. And so the reason why, so if we're looking at one example, so in this case we have the control samples in black and the experimental group in red. And the order, now first time we measure them in the order, so we first measure all the random ones and then measure, all the control ones, and then measure the experimental group. So in this case, if there's no change of sensitivity of our measurement, then it doesn't matter. If we do the randomized or non-randomized, we'll get roughly the same result, that there is no difference. So this is the same case when there's no change in sensitivity. But let's say that we have a linear change in sensitivity, so in this case the instrument becomes more sensitive after a while. We can just by if we don't randomize the order, we get a very low p-value, so there's seemingly a big difference here. But if we would have randomized the order, we would have gotten again a low p-value, although the standard deviations, of course, would have been much bigger in the case, because we have this linear trend. And we definitely don't want this to happen, because we would get the false result. So that's why randomization is important. Another thing is blocking, and blocking is used to control for known and controllable factors. So we want to minimize the effect of variability associated with different things like location, operator, plant, batch, the time of measurement. So so for example, in this case, we have randomized if we have 4 instruments and 4 operators. And in this case, we have a lot of material, so we can run each sample on each instrument once and by each operator once. So now we run each sample 4 times, but always a different instrument operator combination. So then we can also do another, so this was called the randomized complete block design. Then we can also do what's called a Latin square design. So here we're really minimizing the effect of 2 independent factors. So here we have the rows and columns represent 2 restrictions on randomization. So that we have sorry, so I actually it's the same. So this table is wrong. So sorry, it's so here we didn't, we were just looking at 4 instruments. So it shouldn't be that we have 4 operators in this case. So this is just with 1 variable. But we ran each sample on each instrument to see, to make sure that the effect of instrument didn't come into play. Yeah, there's no operator. Yeah, so that should be, that was a mistake to put that in there. So here it's when we look at 2 different factors and then have the restriction on both rows, so that each sample is analyzed on each instrument, and each operator analyzes each sample. And this is so this is a similar thing that from the Sudoku game, where you have the both on rows, and I mean that is has another layer of complexity, but it's a similar thing that you have these restrictions on rows and columns. So then another thing we want to do is replication. So we want to do our experiment several times to look at what the variability is. And there we have different levels of replication that we can do. So we can, usually in proteomics, we talk about technical replicates, process replicates, and biological replicates. So technical replicates would be that you prepare the sample once, and then you just run the mass spectrometer, the chromatography and the mass spectrometer several times on that sample. Process replicate would be that you go through the whole sample preparation process from the beginning for each replicate. And then biological replicate, that you would actually have different biological materials. And it's important to sort of realize the difference of this, because there is in the literature sometimes a bit of a confusion between these different replicates. So people do technical replicates and draw conclusions when they could only draw those conclusions when they would have done biological replicates. That's not that does happen. So, and what's usually nowadays, technical replicates are the error associated with technical replicates is very small in proteomics usually. With process replicates, they can be larger, but still usually both the process and the technical are much smaller than the biological replicates. So the so the main reason for replication is that we want to be able to measure something more accurately, and we want to know the variation. So this just shows why it's, if we have more and more replicates, so these are just 4 different types of distributions that we randomly pick from. And then if we increase the number of replicates, the distribution becomes more and more random, meaning that the standard error of the mean becomes smaller and smaller. So that's the advantage. So we have to do many replicates, we get very much more accurate measurements of the mean, and the advantage is also that we can, if we do replicates, we can estimate the error, which is important if we want to do any type of statistics. So this is an example of a bad experimental design. So if you see here, this is a case where we did either it was a sample taken before or after treatment, and we use different gradient lengths to do the LC, and this is sorted in the time order. And this is, there were 2 different laboratories that this was analyzed in, and these are the patient samples. So for example, here we have patient 35 before treatment, analyzed on a 3-hour gradient in laboratory 1. And then this is the same patient after treatment. That sample was analyzed in laboratory 2 using a 1-hour gradient. So this is actually a real-life example of someone that came to me with this data set to analyze. And it took a little bit of, I mean, I had to ask them several times. This wasn't obvious that this had happened. So I had to ask them what was, and made this table finally. And it really, I mean, it can't be worse than this almost. So pretty much all the lab 1, so the person had moved from one lab to the other. I mean, the person that did the mass spectrometry. But then there was someone left in that lab that still could run a few samples here. But then they changed the protocol from 3 hours to 1 hour. And then they switched back a little bit. But it's, yeah. So, yeah, don't do this. And I'm not going to reveal who did it.
Did you extrapolate anything?
Sorry?
To a complete analysis? Or like
no, I told them to redo it.
Okay.
And they did.
Okay. They wanted to compare.
And this was actually, I mean, as you see, it was quite, took, I mean, several months.
Yeah.
I mean, they probably did other analysis in between, but still, it's not a negligible amount of work to do this. No, they actually agreed to throw everything away and start from the beginning. So just to summarize this part, the experimental design. So as we said, chance and bias is a threat, and it's a very serious threat to the conclusions. And as you saw in the previous slide, an experimental design like that, well, there is a serious chance of introducing a lot of bias that way. We talked a little bit about controllable factors and uncontrollable factors, and how one can sometimes make uncontrollable factors controllable. But we can also use randomization to guard against the unknown and uncontrollable factors. Replication is very important. We can use it to estimate error, and we can get more precise measurements. And we can also should use blocking to control for known and controllable factors. And last time we talked about how we should apply multiple testing when we do large measurements. So in general, the experimental design isn't really difficult, but one has to take time to think about it and pay attention. That's the main thing. There seem to be, I mean, I think now it's getting better, but initially when people were able to generate these large amounts of data, first with microarrays, then with large proteomics experiments and next generation sequencing, people forgot about basic experimental design and sort of thought that, oh, this is magic. But now it's getting better. Yeah. So back to molecular markers. So there are several products on the market, more than we can talk about here, and some are sort of more successful than other. So this is one example called MammaPrint, and so this is a 70 gene signatures to stratify women with breast cancer into lower high risk groups for reoccurrence of the disease. And then one can use that to then be more aggressive with the treatment or less aggressive. And it's definitely worth, I mean, these tests can cost quite a bit of money. So if one can, that's one of the driving for why a lot of people are trying to come up with molecular signatures, that because you can earn quite a bit of money on these. And so, for example, for MammaPrint, so this is a while ago in 2007, they have performed a lot of tests. Another one is Oncotype DX. It's also for breast cancer, so it's 25 genes. And it's to predict whether a woman with localized ER-positive breast cancer is at risk for relapse. So as you see, so a lot of these are assays are quite specific. So it asks a quite specific clinical question. And that's another thing that's very important when one tries to develop a signature, is to choose a good and well-defined clinical question. Because otherwise it's not, I mean, the signature is not really useful. So the other thing is that a lot of the tests can improve both survival and save costs for the healthcare system. So then, if we look at the first very promising proteomic molecular marker was published in 2002. So it had a 100% sensitivity and 95% specificity for early detection of ovarian cancer from serum. So it was it was great because you could just take a blood sample and then do early detection of ovarian cancer. Unfortunately, it turned out to be wrong. And it actually caused a lot of problems for proteomics in general, because when this came out in 2002, everyone invested a lot in proteomics and in clinical proteomics. And then when it turned out that this didn't work, it was proteomics got a bad reputation. And it's still, I mean, now it has recovered from it, but it took many, many years actually to recover from it. So what went wrong? So what happened is that the data was available, so other people started analyzing it also. Then they saw that that there was some problems with it. So one thing they saw, that it seemed that, again, like in the previous example, that the control group and the samples, the ovarian cancer samples, had not been treated the same way, and mainly not been analyzed the same way. So they were groups of samples that the ovarian cancer had been analyzed first, and probably the normals later. So the same thing as the other example. It was. So then it actually. They tried to get it FDA approved, and finally it failed FDA approval because it didn't reproduce in a bigger sample. So, and actually this, there is another interesting thing with this molecular marker, that it wasn't based on a list of known proteins. It was based on a pattern. So it was based on the actual spectrum that you got from the mass spectrum, and just so this and the way.
It was protein independent?
No, but they couldn't. One didn't know which protein, and probably a lot of peaks were many proteins together. So the way that this was done was with a variation on matrix-assisted laser desorption mass spectrometry. So you would mix a matrix with your sample, put it on a surface, and then shoot a laser light, and then look at the ions. And so you didn't have any separation, or they did some little bit of but very little separation. So you would have all the proteins from serum that you would analyze. So it was a very, so you got peaks, you didn't know what the peaks were, and they were probably often many proteins together. And there was an instrument that was marketed to and sold to many hospitals, including NYU had also an instrument like this. So this first 2002 Lancet paper led to a lot of interest, and it really didn't work now that mass spectrometer is gone from the market. So what we need to develop a molecular signature is, first of all, a well-defined clinical problem. So that's the most important. And a lot of studies, at least in the early days, failed on that. Then we need some kind of high throughput assay, and then we need computational statistical methods to analyze it. Then the first thing to do is to get the baseline characteristics of the study. So if we're looking at, so this might be a little bit difficult to see, but what you have here is, for example, age, race, and nodal status, and just clinical variables that we want to sort of match up between the different groups. So, for example, we want the age group to be roughly the same. and this is not so. Uh, Again, so this one should definitely have a table like this in a paper that publishes because I have reviewed papers where the age groups were extremely different between the control group and the disease group. So some people looking for markers in ovarian cancer, and the control group that they had were pregnant women, because it was easy. I think they were looking for markers. If I I'm not sure how they would, but anyway, for some reason it was easy for them to get these samples from pregnant women. So the control group was probably in their 20s or maybe 30s, and the ovarian cancer group were in their 60s. And on top of that, the control group was pregnant. So it was, people do these things because it's convenient to get some type of samples. Oh yeah, so we talked a little bit about how to address bias, and there are many different ways. So I definitely recommend David Ransohov's paper, and he has a whole series of papers, but this is a good one to start with. The bias as a threat to the validity of cancer molecular marker research, where he goes through how to avoid different types of biases. So now if we get to the analysis, we can start with a simpler example, and in this case we have two groups. We have boats and houses, and we want to find a way to distinguish what is a boat and what is a house, and label those in this picture. So one observation is that we have the coastline, and we can make the assumption that all the objects that are in the water are boats, and anything that's up on land is a house. So the computational part would be to find the coastline, and then as always, so then we can, of course, we have two variables only here. So we have the longitude and latitude, and then we can do this classification when we know and remember, with high throughput assays, we don't just have two variables, we have thousands or tens of thousands of variables. So with this, we find an algorithm that best separates these two classes. And then when we look at an unknown object, then we can classify unknown objects. Anything that's up above the yellow line is a house, anything that's below is a boat. But of course, we're going to make mistakes. So the houseboat, this house will be classified as a boat, while these boats that are up on land are classified as houses. And so this is what we're going to run into also with molecular markers. But we have a much more difficult problem, so because 2-dimension is relatively easy, but if we have we can have an enormous amount of dimensions. So if we and so the raw data here, so this would represent the raw data in these different assays, is very big. So we try to then use our knowledge of biology to distill it down to a smaller number of dimensions. The other problem is that we have many measurements, but we usually have very few samples. we don't have, I mean, in a proteomic study with 100 samples is quite large. So you can imagine we have all these measurements, but we only have 100 samples. So we're going to be able, and that's what's in this case called the curse of dimensionality. And the main reason is that it's cheap to measure lots of things, but it's expensive to measure a lot of samples. So we have all sorts of problems. Some methods don't run at all because they're too slow. Some methods could give very bad results depending on the data. And the main issue is overfitting. That we're gonna and overfitting is pretty much mainly that we fit our model very well to the data that we have, but then it doesn't generalize at all. So there actually could be two problems, either overfitting or underfitting, but in our case it's really the overfitting that's the big problem. So here, if we have an outcome and the predictor, this red line here is a pretty good fit to the data. But if we would start with a much fewer points, so we've only measured these points, the blue ones, then if we fit a curve that goes through all the points, that would be very bad for predicting what the other data points are. But if we would just fit this line, we would that would be a much better description of data. But the problem is that it's very difficult to know when we have done an overfitting. And the main reason is that we can, if we have a very large sample set, we can then divide it up into training data and testing data and do quite well with that. But usually, since we have few samples, we can't do that really well. So this is just an example of underfitting. So this data seems to be more probably a threshold and then increases. So here in this case, a line would underfit it, but something that goes like that would fit better to the data. So yeah, so the main problem that we usually have is that we have our signature is too complex, meaning that it fits the training really well, too well, and that it doesn't generalize. But we do want to not have a too simplistic signature, because that's not good either. So the general problem is that when we have, again, few samples, many measurements on each, it's really easy to develop a predictive model. And even we often can really believe that it's a good signature, even when it's not. So there are many, and we saw one example of the overcheck, which is looked very promising initially, but didn't get approved by the FDA. So one approach to this is to an example is support vector machines, which is a machine learning approach to extracting the molecular signature. So here in this case we have, for example, two groups. One normal and one cancer patient group. And then we're looking for, they're described by two genes, gene X and gene Y. And we want to try to find how to divide these two. And as you see in this case, it's pretty straightforward if we draw a line here. And that's what the support vector machine would do, that it would draw a line that maximizes the gap. And in many dimensions, then, of course, it would be not a line, but a hyperplane. But again, it would look for the largest gap. And then there are tricks. So, for example, in this case, there is no line that can separate these two groups. But then what one can do is to add transform this into a higher dimensional space so that one can separate it with the surface in this case. And that's so this is what one can do with support vector machines. And so this is a very flexible way to approach this problem of finding decision surfaces, hyperplanes between different groups. So as we said before, the best case is if we have a large number of samples. So we have a data set, and we divide it into a test set and a training set. So first we train our model on the training set, and then we test it on the test set. But this is unusual that we have this amount of data. So another approach would be what's called n-fold cross-validation. So we have our data set. We divide it up into a small test set and a larger training set. And then but we do this many times. So that, and each time we train our model again and test it. Again, here, train it and test it on a new test set. And this way, and you might have heard of, so usually people use either 5-fold or 10-fold cross-validation, depending on the data set size. And that works reasonably well, but definitely not as safe as having a very large sample. So one can really have a separate training set and a separate test set. So then there are several problems that we run into. One is that we can, because of this case again, that we have few samples, many measurements, we can extract many different signatures that would work equally well in our data set. But then it's very difficult to predict out of these set of which ones would best generalize. And as we discussed, often we have very poor experimental design that we've seen examples of, but that can of course be avoided by thinking about it properly. Sometimes there is no predictive signal. Sometimes, even though we've done all these measurements, there is really no, we haven't measured the right things. So there is really nothing we can do with it. And preferably we should recognize that and then redesign the experiment. But it's not always easy to recognize when that's the case. Then we talked about reproducibility and validity. Then there are other problems also that we discussed in the past. And one thing is, of course, that it's much easier to publish something sensational. So people are always pushing their analysis to find something that really could be make a very big difference. So just to summarize, so molecular signatures is very critical in precision medicine, and there are already a lot of signatures developed, but there are need for many more and better signatures. So they're already now used for treatment, but there's definitely a lot of room for growth. And then we often have this problem with multiplicity, and there are still some very interesting problems for computational analysis of omics data and how to do that best. It's definitely not a solved problem, and people are constantly developing new algorithms. So that was everything I wanted to go through today, and